enjoyed the lesson, right? Of course, it's really very surprising that how such a small cell contains these many organelles, and they all do wonderful activities as we do in our daily life. Bindu is back with finally part 5, class 9th biology, to make you understand the difference between cell wall and cell membrane, cytoplasm and protoplasm. And also various interesting activities. Learning objective. Students, now after this class, you would be able to recognize the difference in plant and animal cell. You could be knowing very well about cytoplasm and protoplasm without any confusion. And also, you could easily identify all the organelles present in the cell under microscope. Before going to this lesson, focus on these keywords. Protoplasm, cytoplasm, cytoskeleton, methylene blue, microscope, permeable membrane, safranin. Take a quick look. Let's complete the lesson with this table. Cell wall, seen only in plant cell. Cell wall is made up of 3 layers called primary layer, middle lamella and secondary layer. Plant cell wall consists of tiny cellulose fibers called microfibrils made up of pectin, cellulose and hemicellulose glued together by a mixture of polysaccharides. Middle lamella is a jelly-like substance made up of calcium and magnesium pectate. Protects the cell from physical injury. It maintains the shape of the cell. Regulates the flow of information between cells. Provides protection against pathogens. Plasma membrane. Selectively permeable membrane. Cell membrane. Semi-permeable membrane. Differentially permeable membrane. It is made up of proteins and phospholipids. The arrangement of protein and lipid in 2 models. Sandwich model means lipid-protein-lipid and fluid mosaic model means lipid float in the protein layer. It allows only selected particles into the cell like nutrients or some salts. Presence of lipids and proteins provide flexibility to plasma membrane. Protoplasm. It is a jelly-like, colorless, transparent and viscous living substances present within the cell wall. Protoplasm is bounded by a thin membrane called plasma membrane. Its thickness is 7.5 nanometer. Carbon, hydrogen, Nitrogen and oxygen are the major elements constituting about 95% of the protoplasm. The remaining 5% consists of calcium, phosphorus, chlorine, sulfur, potassium, sodium, magnesium, iodine, iron. It is responsible for various living processes. It was believed that cells were containers of protoplasm. Cytoplasm. The part of cytoplasm extending from cell membrane to the nuclear membrane is cytoplasm. The cytoplasmic matrix is homogeneous and shows a cytoskeleton formed of microtubules and microfilaments. It is transparent, viscous, colorless fluid composed of chiefly 85% to 90% water, 7 to 10% protein, 1 to 2% fat, remaining all with other inorganic materials. Cytoplasm moves all over the cell. Cytoplasmic movements are of two types. Rotation, move either in clockwise or anticlockwise. Example, in the cells of hydrophytes. Circulation, moves in zigzag manner around vacuole both in clockwise and anticlockwise. Example, hair cells of rhodoscula. Eyepiece. The eyepiece or ocular magnifies the primary image produced by the objective. Body tube separates the objective and the eyepiece and assures continuous alignment of the optics. Revolving nose piece to rotate those objective lenses and help to change the magnification power. Objective, responsible for primary image formation and plays a central role in establishing the quality of images that the microscope is capable of producing. Condenser, gather wavefronts from the microscope light source and concentrate them into a cone of light that illuminates the specimen with uniform intensity over the entire view field. Fine adjustment knob, used for initial focusing. Coarse adjustment knob allows for quick focusing by moving the objective lenses or stage up and down. Inclination joint, it will tilt your microscope back for more comfortable viewing. Observe plant cell under microscope. Take real leaf. Tear the leaf in a single stroke. Observe it against the light. Take a small piece of leaf peel with light colored transparent portion. Put it on slide and put a drop of water on it. cover it with cover slip and observe the lighter portion of leaf under the microscope. You observe the following. Now put 2 to 3 drops of dilute salt solution on the membrane and leave it for 5 to 10 minutes. Observe it under microscope. Observe nucleus under microscope. Aim to observe the nucleus in cheek cell. Material required: a toothpick or ice cream spoon or spatula, glass slide, cover slip, watch glass, needle, blotting paper, 1% methylene blue, normal saline, glycerin, microscope. Procedure: wash your mouth and scrape a little of the internal lining of your cheek inside your mouth with a clean toothpick or spatula or ice cream spoon. Place the scraped material in a watch glass containing a very small quantity of normal saline. Then place the material on the glass slide. Put a drop of methylene blue and wait for a couple of minutes. Put a drop of glycerin over it. Place a cover slip. Tap the cover slip with the blunt end of the needle so as to spread the cells. Wipe off the extra stain with a fine cloth or blotting paper. Observe the temporary mount under low and high power of microscope. Dark colored spherical or oval dot like structure near the center of the cell are seen. This is nucleus. Precautions: do not scrape the cheek too hard as it may injure your buccal mucosa. Scrapped material should be spread uniformly on the slide. Excess stains should be drained off. There should be no air bubbles under the cover slip. Observe mitochondria under microscope. Let us do this activity with onion peel. Make a fresh solution of Janus green bee in a beaker. Mix 200 mg Janus green bee in 100 ml of water. Take a watch glass, pour some solution. Put the onion peel in this solution and keep it for about half an hour. Keep a piece of onion peel on the slide and wash thoroughly with water. Cover the slide with a cover slip and observe it under microscope at high magnification. Cylindrical grains scattered in the cytoplasm. These are the mitochondria. Observe chloroplast under microscope. Take the peel of Rio leaf and mount it in the water drop on a slide. Observe it under compound microscope. You will observe small green granules called chloroplast. They mainly contain green substance called chlorophyll. Observe vacuole under microscope. Take the leaf or stem of any succulent plant like the torch cactus. Take a thin peel from the leaf. Take thin cross section of stem of cactus in a watch glass containing water. Stain it with dilute surfactant solution. Observe the section under low and high power microscope. The large empty spaces present in the cell are vacuoles. Let us see all the scientists who are involved in this lesson. Robert Hooke. Cells were first discovered by Robert Hooke in 1665. He observed the cells in a cork slice with the help of a primitive microscope. Leeuwenhoek in 1678, with the improved microscope, discovered the free living cells in pond water for the first time. It was Robert Brown in 1831 who discovered the nucleus in the cell. Purkinje in 1839 coined the term protoplasm for the fluid substance of the cell. The cell theory that all the plants and animals are composed of cells and that the cell is the basic unit of life was presented by two biologists, Schleiden in 1838 and Schwann 1839. The cell theory was further expanded by Virchow in 1855 by suggesting that all cells arise from pre-existing cells. With the discovery of electron microscope in 1931 by German scientists Ernst Ruska and Max Knoll in 1940, it was possible to observe and understand the complex structure of the cells and its various organelles. It is a modern microscope. It can magnify approximately 2000 times better and can distinguish the objects about 0.0004 inches across.